biotechnology. Okay, Suyang, yeah. Okay. So for the biological, you learn about how you use the bacteria to, how you use the bacteria to produce another kind of useful products. So the products will bring benefits to the human life. Okay, you can refer to this one, which is this one, the chapter 21. Okay, chapter 21, the important keywords. Okay, so at this one, whenever you do the revisions, right, you look about the notes, and then you are not sure which is the important point for each chapter. So you can refer to this one. Okay, you can refer to these important keywords, and then I have uploads the every chapters for the for this one important keywords at the Google Classroom, and then you can access this. And then when you read through the notes, right, you can see some of the questions I put at the start, because those questions are being come out in the exam before. So that's why you need to be aware of it. At least you need to ensure you already know how to answer the question by using the correct keyword. Because IGCSE exam, right, they are really focused on the keyword. You need to answer your question with the correct keyword, which is already stated in the answer scheme. So if you didn't answer with the correct keyword, right, even though your answer sounds correct, but you will not get the mark, because the examiner, they will mark based on the keywords. Okay, so that's why you need to take note of the yellow highlighted or the red color keywords. Okay, so we start our lesson now. We can use the bacteria to do, to make a lot of useful substance. For example, because bacteria itself, right, they are capable for producing the complex molecule. Yeah. For example, right, the bacteria are added into milk to produce an enzyme, so they will turn the milk into the yogurts. Okay, so usually this kind of bacteria, right, they can produce the lactic acids. Yeah, because yogurt is always taste sour, right? I think you drink yogurt before. Yogurt tastes sour, right? Why yogurt tastes sour is because yogurt contains the acid called as lactic acids. And then this acids, right, is produced by the bacteria. It is being produced by the bacteria. What kind of bacteria? Have you heard of lactobacillus, which is this kind of bacteria? The bacteria which produces lactic acids called as lactobacillus. Bacillus is referred to the shape of the bacteria. Yeah. So this kind of bacteria, right, they are being found in a lot of food products, include in the yogurts and also in the, in the Yakult, okay, Yakult that they will drink before. Yeah. So why they use bacteria but not other kind of living organism? Because bacteria are so useful. As bacteria, they do what? They do the asexual reproductions. Yeah. So they can produce, they can reproduce rapidly in a very short time. They can produce a large amount of the progenies. Okay, so that's why the amount of chemicals, which is the products they produce, can also rapidly increase. So for example, you grow the lactobacillus bacteria in that mediums. So the lactobacillus, they will grow, okay? They will grow and increase the bacteria number. So whenever you increase the number of bacteria, you can increase the product which is being produced by the bacteria. So you can increase the productions, you can increase the amount for the production of the lactic acids by growing the lactobacillus. So here's an example. Okay, and then other than that, right, the bacteria, they have the ability to make the complex molecule. Yeah, and then why they are using bacteria is you use the bacteria, there will be no ethical issue. If you use the animals or humans, especially when you use the humans to do this kind of experiments, you will raise a lot of ethical issue. But use bacteria, you will no issue. So that's why you can just grow them in a large number in the laboratory, and then no one will question you. Yeah, and then another thing is bacteria, they have useful structures, which is only found in bacteria, not in other living organisms. So the structure is called as what? It's called as plasmid. You should remember plasmid. Okay, before that, I want to ask you, at this way, bacteria is eukaryote or is prokaryote? Hi, Suya, Eden. bacteria is prokaryote or is eukaryote?
Prokaryote.
Yes, your answer is
Prokaryote.
prokaryote or eukaryote?
Prokaryote.
Prokaryote, okay. Thanks. Suyang, are you okay? You are still working.
Yep.
You can listen in. Okay. You can listen back to the recording, okay? Yeah, it's prokaryotes. Well done. Okay, you still remember it. So bacteria is only the one living organism they are classified as the prokaryote. Okay, it's a prokaryote. Other than that, it is the animal cell, plant cell, the fungi, and then the protista. They are all classified under eukaryotes. You know how to differentiate the prokaryote eukaryotes? This is one of common questions come out in exam. So how you're able to differentiate is the eukaryotes, they have the nucleus. Eukaryote, they will have the nucleus, okay? Nucleus with the linear DNA, linear form of DNA. And also they will have the membrane-bound organelle. And then prokaryotes, they don't have a nucleus, okay? They don't have nucleus. They have what? They have a circular form of DNA, and also they don't have membrane-bound organelle. Okay, yeah, they have a ribosome. They have a ribosome, and then they have a plasmid. Yeah. So plasmid is a kind of DNA. In the prokaryote bacteria, they have two forms of DNA. One is called circular DNA, another one is called plasmid DNA. So plasmid are referred to the small circular loop of DNA, which can be ideal way of transferring the DNA from one cell to another during the genetic manipulations. So what is meant by this is referred to this one. Okay, sorry. Let me show you a process. I think you can refer to this note. The process. I could put a picture here. Yeah, here is a picture. Yeah, okay. You can refer to this picture. This is I put at the chapter 21 important keywords. So at the so you can have a look for this picture. Okay, what is meant by genetic modifications or genetic engineering? We call it 基因改造. How we able to modify the gene compositions? So, for example, right, if you want to insert part of the human gene, for example, in this picture, they show that they want to insert the human insulin genes, okay, just that gene, okay, the specific gene into another living organism, which is they want to insert into the bacteria. Yeah. So what they can do is first they will isolate the bacteria, okay, so they will isolate the bacteria, and then they will isolate the plasmid DNA from the bacteria. Means that they will just take out the plasmid DNA from the bacteria. They just take them out. So with the plasmid DNA, right, they will use the enzyme. Okay, they will use the enzyme to cut to cut a regions, okay, a small regions on the bacterial plasmid. So as you can see this picture, right, this is a plasmid just isolated from the bacteria because bacterial plasmid. So it is a circular form of DNA. Requires loop of DNA. So you want to insert the human insulin gene later into this bacterial plasmid. So now you don't have space. So that's why you need to use the enzyme, because what? Restriction enzyme. So restriction enzyme is just like a scissor. You just cut regions on the bacteria. Yeah, maybe they just cut here. Okay. They just cut here, and then they will expose. Because what? The sticky end. Yeah, so this is called sticky end. So later, they will use the same enzyme also, which is also the restriction enzyme, to cut on the human chromosome. So they want to isolate the particular genes, right, which is the human insulin genes, from that human chromosome, because chromosomes are super big. On the chromosome, right, there will be thousands of genes located on here. So that's why they need to target on that specific locus on the chromosome to isolate that specific gene, which is in this case, it is the human insulin gene. So they also use the enzyme to isolate that particular gene out. Okay, so now they already isolate out the genes and then with the sticky end. Yeah, so you can see, right? Yeah, you can have a sticky end. So that's why now you need to join. You need to use the another enzyme. So you can join the human insulin genes into the bacterial plasmid. Yeah, so that's why the name of enzyme is called as what? DNA ligase. Okay, so here is the enzyme. So DNA ligase just acting like the glue. Okay, because this glue, they can help to stick the bacterial plasmid and the human insulin gene together. So now the plasmid will transform, become recombinant plasmid. What is known as recombinants? Recombinants are referred to that plasmid, they carry the foreign gene. Okay, like in this way, in this case, right, they carry the human insulin genes on that bacterial plasmid. So they become recombinant plasmid. So later they will insert this recombinant, later they will insert this recombinant plasmid into the bacteria. So you can see in the picture label number 4. Yeah, so now they just insert this plasmid into the bacteria. Later, they will grow that bacteria into the medium, which is they will provide the bacteria with the optimum temperature, the pH, the nutrients. They will supply to the bacteria. So they'll just grow the bacteria in the medium or in the culture flask. So the bacteria, they can do the asexual reproductions, which is a mitosis. So they can increase their number of the offspring in a short, in just a very short period. They can produce a lot of bacteria by the asexual reproduction. So by this way, they can produce a lot of products. So in this case, because you are actually inserting the human insulin genes into the recombinant or into the plasmid of bacteria. So later the bacteria, they will carry out their own process, which is a transcription and translations. So they will use the genetic information from the human insulin genes. So they'll use this information to make the products. So in the product will be the human insulin proteins. So you can see that here is a human insulin. The insulin proteins. So insulin proteins, okay, is produced by the recombinant bacteria. So that's why bacteria will express the genes and then they will produce a lot of human proteins. So later, the human proteins, which is a product, they need to be collected and then they will need to be purified. And then now you can use the human insulin proteins for your the purpose to treat the diabetes patients. So here is the process. So now you understand why they want to use bacteria, because it's very, very convenient for them to do those kind of experiments. Okay, so that's why, apart from that, right, what kind of products are related to biotechnologies? So now we call it what? Biofuel. Okay, what's mean by biofuel? Biofuel, with the term of bio and the fuel. So they are actually using the biological organism to make them become source of fuel. Yeah. For example, yeast. Okay, yeast is a unicellular fungus. So what the yeast will do is they will use the sugar as the food source. So yeast, they can do what? They can do the anaerobic respirations. I hope you still remember about the equations for the anaerobic respiration. Yeah, the equations, right? Yeah, which is this one. C6H12O6, and then you convert, become the ethanol, C2H5OH. So, and then you produce the carbon dioxide, CO2. Okay, so this is the equation for the anaerobic respiration, which is an incomplete breakdown of glucose. Okay, because the glucose, they will not able to break down completely in the absence of the oxygens. So there's no anaerobic respiration in the yeast. So that's why the scientists, okay, they are actually using the yeast. So they use the yeast a lot in the fermentation process. So by this process, they can produce a large amount of the products. So which is the ethanol and carbon dioxide. Yeah, so ethanol, they can be used to make what? Alcohol drinks. And then carbon dioxide, they can be used to use in the, we call as bread making process, okay? Because carbon dioxide, they can raise the dough level, right? Okay, so this example it, yeah. So ethanol produced by the anaerobic respiration in the yeast, they can be used in production of biofuel. So in country such as Brazil, biofuel is replacing the petrol as a fuel for the vehicle. Yeah, because you can produce ethanol. Now, ethanol are same to the something like petrol because both are known as a non-polar solvent. Yeah, so that's why you can use this ethanol as a source of the fuel for the vehicle. Which is they will incorporate this ethanol component into the biofuel, into the petrol. And then biofuel productions using the yeast, right? They can do what? Okay, plant material is broken up into smaller pieces and mixed with yeast. Okay, because yeast is also known as the decomposer, one of decomposer. You should remember, decomposer made from the bacteria and the fungus. So yeast is belong to the fungus. So yeast cell, they use a plant material as a substrate in the anaerobic respirations and produce ethanol as the products. So let's have a look. So later, the liquid is separated from the remaining solid and water is being removed, leaving the concentrated of ethanol. So by doing these anaerobic respirations for the yeast, they are actually process the fermentations. So this fermentation, they can produce a large amount of the ethanol. So this ethanol can be purified out, okay, for other usage. So you can see later the ethanol, they can blend it with the petrol, or they can use its own as fuel. So that's why it is possible to use the waste part of the crop plant, such as stalk or outer leaf, in the biofuel productions. But the crop may also be grown specifically to be harvested for making ethanol. So it means that you can harvest the plant, which is a waste part of plant. Because usually we think that, oh, okay, this kind of plants, they already, the wild plants or the plant already died, okay, with a dry yellow leaf, right? You think that they don't have any kind of usage anymore. But actually, you can process the waste part of the plants, become the source of the ethanol. Yeah, you can harvest them for making the ethanol. Yeah, so this will cause a concern because there's less land available for local products to grow foods crops need for the survival. Okay, so here's this example of usage for the biofuel. So just remember that biofuel by the yeast through the fermentation process, which is the anaerobic respirations, to break down the glucose, produce the ethanol and carbon dioxide. So ethanol later, they can be used as a source of the biofuel. Okay, so how you're able to how you're able to perform this yeast fermentation to produce the ethanol, so you can carry out the anaerobic respirations. Yeah. And then another thing is bread. Okay, see, used in the bread making process, because yeast, they will respire anaerobically, and then they will access to plenty of sugar, which is they will just break down the sugar in the absence of oxygens. So that's why when the yeast are being mixed with flour and water, they will respire anaerobically, and then they'll produce a large amount of carbon dioxide. So this carbon dioxide is very useful for them because they can make the bread dough rise. So whenever you see the surface of the bread, right, it's not very smooth. They will have a lot of small hole. So this small hole is evidence that the yeast carry out the respirations. So they will produce a carbon dioxide. So this carbon dioxide, they will produce a hole, okay, within the dough. Yeah, so this is the important point, okay? And just yeah, the carbon dioxide produced by the yeast is caught in the dough. So they're causing the bread to rise. Okay, they can increase the level of dough. Means that 面团会膨胀. And then I think is fruit juice productions. Okay, how they can use the biotechnology, which is they actually use an enzyme. They added this enzyme into the fruit juice. So the enzyme name we call as pectinase. Okay, pectinase. So because the fruits is belong to the plant cell, and then now we talk about the cell wall of the plant cell. Okay, cell of the plant cell. So at those what made up the component of the cell of plant cell? So remember what is the structure? It's a kind of polysaccharide. What made up the component of plant cell? Wall.
Cellulose.
Yes, cellulose. Okay, so remember. Well done. Yes, it's cellulose. Okay. Not only cellulose. Actually, they also have the pectin as well. But this pectin, right, is not commonly asked in exam. Exam they will ask you cellulose only. Yeah. So these pectins also become one of the component in the plant cell wall. It's very hard and rigid structures, just like in the cellulose. So what they're actually doing is they will just add this enzyme pectinase. So this pectinase, right, they will just break down the pectins. So you can see it's a kind of enzyme help to break down the pectins in the plant cell wall. So once the pectin is being broken down, the cell wall is being break more easily. Yeah, so they just break the cell wall. So once they break the cell wall, they can release more content which is inside to outside. So they'll be allowed more fruit juice can be squeezed out from the fruits because the cell wall already been break down. Okay, so by this way, right, adding the pectinase to the fruits, you can help to produce a clearer juice, okay, with a larger volume. Okay, because larger polysaccharide like the pectins, they can make the juice to appear cloudy. So once the pectins are being digested or broken down into small molecule, the juice become much more clearer. Yeah. So means that the appearance of juice, right, they will look more nicer, and then you actually have a you can get more fruit juice. You can, because you will see the pectinase, right? You can allow more fluid inside release to outside. Yeah, so that's why you can increase the volume of fruit juice also by using the pectinase. Okay. Yeah, that's all. Yeah, because if you use a traditional method, which is chopping the fruits or you're squeezing them, you can release a lot of juice, but this will not break down the cell wall. So this will still cause some loss of the juice productions. So that's why you need to use a pectinase. It will be very effective way to release more fruit juice instead of you chopping them or you squeeze them. Another thing is in the biological washing powder. I think you got help your family to wash your clothes, right? So the detergents, right? The detergents washing powder is made from what? It's made from the enzyme. Yeah. So many stains on the cloth are the organic molecule. For example, the oil from the skin, the protein from the blood. So this is related to the female. Okay, female. Fats, proteins from the fruits. So this will cause your clothes, right, appear very dirty. So how you can wash your clothes is by using the detergents. So usually the detergents, right, okay, they contain the salt. They can remove some of stain when mixed with the hot water, but they can take a lot of time and effort and very high temperature to remove the stain entirely. So that's why usually they use a biological washing powder. Okay, C21 now, we call this one C21. So this washing powder, they will contain the enzyme. So this enzyme, just similar to the digestive enzyme they will learn before, which is the lipase, protease, the carbohydrates, yeah, which is produced along your alimentary canals to break down the large food molecule. So they just incorporate this kind of enzyme into the biological washing powder. And then usually the enzyme is what? Lipase and protease. Okay, so that's why those enzymes, they can break down the large insoluble molecule such as fat and proteins into small and soluble, so they can dissolve in the washing water. So that's why it will be effectively, they can remove the dirty stain from the cloth. Yeah, and also those kind of enzymes, right, they are effective at the lower temperature. So that's why even though you wash your cloth with the cold water, but it's still effective because less energy has to use to wash the clothes to get them clean. Yeah, as the washing water does not need to be heated to the higher temperature. Okay, so the point for this paragraph just showing that in the biological washing powder, there will be two forms of enzyme. Okay, one is protease, one is lipase. Yeah, these are protease and lipase. It can help to break down the protein and the lipid stain on the cloth. Okay, so they'll carry out what? They'll carry out the, okay, that's one enzyme. They will do physical digestion or chemical digestion. Which one? Physical or chemical?
Chemical.
Yeah, it's chemical. All right, it's chemical. So that's why when you write the sentence, you should be very aware because you are writing about chemical digestion, right? So that's why you need to use the words large. See, insoluble becomes small and soluble. Chemical digestion refer to they can change the chemical property of the substrate. Okay, so from the insoluble becomes soluble because if you compare to the physical digestions unable to do this. They unable to convert the insoluble becomes soluble because the physical digestion, right? For example, okay, chewing of food, churning action of stomach. Okay, usually these two. Yeah. So what they can do is they will just chop down the food into small pieces. So that's why they can just break down the food, right? From the large particle becomes smaller particle. But the molecule will still remain as insoluble because it is physical. Okay, another thing is we talk about the lactose in the milk. Okay, so human baby are born with the ability to produce the lactase, so the enzyme will break down the lactose. So whenever you are still a baby, right, you will just drink milk and then you can survive. Because you will have a lot of lactase, yeah, produced. Okay, in the certain area of world, many people lose the ability to produce lactase as they get older. So this means that they can become lactose intolerant. So they can react badly to the lactose in the milk and products made from the milks. Lactose intolerance, I think you heard this term before, right? Which is some of the people, right, because their digestive tracts, which is from the ileum, yeah, eat from the ileum. Ileum produce a lactase enzyme, right? So don't know why, but their digestive tracts, right, they produce a very less amount of the lactase. So that's why whenever they drink the milk inside, they consist a lot of lactose. So those lactose will not be break down, okay, because there's not much lactase. So that's why they will have lactose intolerance. Okay, so what is the symptom for the lactose intolerance? So it means that whenever they drink the milk products, right, they will have the we call it indigestibility, 消化不良. So that's why they will have diarrhea. Okay, they will have diarrhea because they are unable to digest the lactose. Yeah, see, symptom of the lactose intolerance includes nausea. Nausea is 恶心, okay, 恶心. 恶心 means like you feel like you want to vomit. That kind of feeling called nausea. Flatulence is called what? Flatulence is something like flatulence, okay, is something like your along with digestion tracts, right? Okay, you produce a lot of gas. So that's why 就是放屁的意思. This is called flatulence. And then diarrhea, yeah, diarrhea is called as a watery diarrhea. Yeah, because their digestive system is being upset by the lactose. So just remember that whenever you eat any kind of food products, right, and then your body unable to digest them well, you will have diarrhea. Symptom of diarrhea. So milk can be made the lactose free by adding the enzyme lactase to it and leaving to stand for a while to allow the enzyme to break down the lactose. So that's why the scientists, they will come out with the lactose free milk. Okay, which is they already added the lactase enzyme into this lactose milks. Okay, so the lactase, they will convert the lactose become what? Okay, lactose is a kind of the lactose, right? It's belong to the, we call it yeah, lactose is belong to the disaccharide. So that's why you break down this lactose, which is the disaccharide, become the monomer, which is the glucose. Okay, so remember? Yeah, so that's why you break down them become glucose and galactose, which is a monomer. So that's why the person with a lactose intolerance, right, they can drink this lactose-free milk because inside these lactose-free milks, they already have glucose and galactose. No more lactose because already been broken down by the lactase. And then another thing we talk about this are penicillin productions. Very interesting things. What's that penicillin? Have you heard about 青霉素? Penicillin is a kind of antibiotic. In why it called 青霉素? It's very common prescribed antibiotic in the medical field. But I think nowadays they get a lot resistant toward this kind of antibiotic, penicillin. So penicillin is the first antibiotic being produced. So the story is just like this. Okay, once upon a time, there's a scientist known as Alexander Fleming. So what they do is they do the experiments, right? And then they left the petri dish in the lab. And then in the next day, right? I think it's next few days, okay, when they look back the petri dish, right, they discover a thing, which is okay, something like this. Let me show you the picture. They discover this kind of thing, the penicillin. So that's why they actually accidentally discover this penicillin mold. Penicillin is a kind of fungus. Okay, it's a kind of fungus. Penicillin plate. Yeah, see, this is the penicillin. It refers to the fungus. But this fungus, they can kill the bacteria. So this fungus can kill the bacteria. So they will come out in this thing. How do I show you? We call it what? Inhibition zone. Okay, this one, inhibition zone. So you can see, right, here is the plate. Okay, let me show you the plate. Which is the plate they grow the bacteria. Okay, so this is a plate they grow the bacteria. And this green color things, right, is a bacteria. But what happens if you can see, right, here will be the clear zone. Here will be clear zone. Clear zone. Why? It's because those clear zone, right, they will have the penicillin fungus grow on it. So the penicillin fungus, they can kill the bacteria surrounding them. So that's why they can appear clear zone without any bacterial growth surround the penicillin. So he noticed that some of bacteria they have left in petri dish has been killed by the naturally occurring penicillin mold. So the penicillin will produce a chemical. So this chemical they can kill the bacteria. So they can prevent it from being infected by certain type of bacteria. So just remember, penicillin mold is a kind of fungus. So this fungus, they can kill the bacteria. So they are known as antibiotic. Yeah. So penicillin is an antibiotic that interfere with the building of bacterial cell wall. Okay, so how penicillin fungus kill the bacteria? How they can kill them? So just remember that bacteria, they will have a cell wall, right? And then what's the name of bacterial cell wall? So remember the name of bacterial cell wall? Peptidoglycans. Okay, this one, peptidoglycans. Yeah, so this peptidoglycan, which is the component surround the bacterial cell wall. So bacteria has cell wall just like in the fungus, in the plant cell, okay? Just different components. So how the bacteria able to kill by the antibiotic? The most common way is the antibiotic, they just targets on the bacterial cell wall. So like this, right? See, this is the cell wall of the bacteria. And then here will be the penicillin. So penicillin, they target cell wall. So what they can do is they will just stab, just like someone use a knife to stab on your heart, and then you will die. Yeah. So because penicillins, they will just puncture a hole on the bacterial cell wall. So that's why it will cause a rupturing a cell. So see, because there's a hole on the cell wall, right? So that's why the inside the cell contents will be released out. Yeah, will be released out. So that's why the cell will rupture and die when it tries to grow. So this means that how penicillin kill the bacteria by targets on the bacterial cell wall and cause a lysis. So this is called what? Lysis of bacteria. You can write it down, and then you can write this as one of the keyword in the exam. They cause a lysis of bacteria, so the bacteria will die. Lysis refer to rupture, 爆炸. So that's why because penicillin is very useful, playing as role as an antibiotic. So antibiotic they can kill a lot of the disease causing by the bacteria, right? So that's why scientists they will grow a large amount of penicillin. So how they able to produce a large amount of penicillin is they will just grow in the fermenter. Okay, fermenter. And then another thing I forgot to tell you is the fungus, right? So you need to be aware of the spelling. The fungus name is called what? Penicillium. Okay, penicillium. And then the chemical they produce to kill the bacteria is known as what? Penicillin. So the penicillin is the name of fungus. Penicillin is a chemical produced by the penicillium. So that's why it is the penicillin. We want this penicillin, okay, because only this penicillin, which is this kind of chemical, they can kill the bacteria by target on the cell wall, causing the lysis of bacteria. So you can grow a large amount of penicillium fungus in this fermenter. We call it what? Biofermenters. Okay, so and then you need to know each of the components in the biofermenters. Yeah. So you can see, right, steaming in. So this steaming in, which is because they want to ensure this biofermenter are being provided in the optimum temperature, optimum pH. So that's why you need to steam it. Okay, you need to steam it. And then another thing is this steaming in, right? They can help to kill the bacteria or any kind of pathogens which is inside the biofermenter. So they can make the product become more purified, more clear, without being contaminations by other kind of products. And then you need to provide them with nutrients, and then the water jacket. Okay, so this water jacket, right, is referred to allow the cooling water in and then cooling water out. So that's why there will be a form of cool water, okay, flowing around this biofermenter. So they can help to cool down, okay, at least cool down the temperature, because they just worry that inside the temperature become very hot. If the temperature inside become too hot, right, too hot, it will affect the production of penicillin by the penicillin also. Because we just want to have the optimum temperature, not the temperature become too hot. And then you need to provide them with the air, okay, the air which contain the oxygens, because it is a kind of the aerobic respiration process. Okay, it is an aerobic respiration process. So that's why you will need to provide it with the oxygens, fresh air. Okay, and then you can see this one by one. So fermenter are the container used to grow or culture any kind of microorganism like the bacteria or fungus in a very large amount. And then this can be used for many biotechnology process like producing genetically modified bacteria and penicillin mold. They can produce the penicillin. Yeah, so this is a condition, okay, and then this I put star here because you need to memorize, because this is a very common questions come on the exam. So you need to provide them with the aseptic precautions. Okay, what's my aseptic? Aseptic means it's very clean without any dust, okay, without any microbes, because aseptic. So that's why usually aseptic, right, they're being used as a conditions like they perform the surgery, 做手术的时候血很干净, so that's why they call it aseptic. So that's why you need to use a steaming, steaming which is referred to what? 水蒸气, okay, steaming. So that's why fermenter is cleaned by steam to kill the any kind of microorganism, and then they can prevent the chemical contaminations, which they will ensure the desired microorganism to grow. Okay, so that's why they use a steaming. They use a steaming to kill any kind of possible pathogens to ensure the aseptic conditions for the products. And then you need to provide with nutrients. Nutrients, usually you provide them with the glucose, okay, the source of glucose, so they can, so that's why the penicillium fungus grow inside. They can do the respirations to release the energy for growth, and they can use the nutrients also to do the reproductions, because reproduction also require nutrient input, even though it is an asexual reproductions. You need to provide them with the optimum temperature. Okay, so that's why you need to monitor the temperature with the props. Prop is referred to a kind of sensor device. Okay, they're just placed inside the biofermenter. So if there's any change in temperature out of the range, so there will be alarm setting. Okay, yeah. So that's why the technical will come to repair it. So that's why they need the prop. So prop is just like detect any detect the range in the temperature to prevent temperature become very too high or too low. And then they can maintain using a water jacket. So this is to ensure the optimum environments for the enzyme to increase the enzyme activity and prevent denaturation. Because just remember that when the penicillin fungus grow inside the biofermenter, it also known as a kind of enzyme catalyzed reactions. So it will need the enzyme help them to do the works, okay, to break down to produce a large amount of penicillin. So that's why enzyme is very sensitive to any change in the pH and temperature. So that's why you need to provide with the optimum temperature. So usually the temperature will be around 37, 38. So if too high temperature, it will cause a denaturation of the protein itself. And then you need to provide it with optimum pH. pH is monitored also using the prop to check whether it's an optimum value of the microorganism being grown. Yeah, so that's why you can adjust the pH using acid and alkaline, which is you can add more acids or alkaline to get the pH that you want. And then C is a aerobic respiration. So that's why you need to provide the biofermenter with the sufficient oxygen. And then this one, agitations. So they're usually stirring paddles, okay? Stirring paddles. Because stirring paddles, right, they can ensure the mixture being mixed uniformly. So you can see here is the stirring paddle. So they're just like rolling around. Something like that, just rolling around, okay? Rolling around. So they can ensure the mixture inside being mixed equally, okay? Yeah, so because they want to ensure the temperature, pH, nutrients, and the oxygens are all distributed evenly throughout the fermenter. So this is to maximize their productions. And then the waste. The contents are filtered to remove the waste created by the microorganism. How they can remove the waste? Okay, so they will have this one. Okay, outlet of product. So by this outlet of product, right, which is after the fermentation process end, okay, they can remove away the products. Yeah, like this. So this biofermenter or fermenters, they are being used to produce the products such as the penicillin, okay, penicillin, and also grow the bacteria inside, which is the recombinant bacteria. For example, the recombinant bacteria, which they carry the human insulin gene, they can grow inside also, so they can produce a large amount of the insulin proteins later. So this fermenter is very, very useful. You need to study, okay, study what's the optimum condition being provided for those organisms. So that's why they can grow well, okay? They can grow well, multiply rapidly, so you can get the products that you want. Okay, so here's the conditions. And then now we'll talk about mycoproteins. Mycoproteins is they are referred to something like the fungus. Yeah, so mycoproteins is this one. The fungus Fusarium is cultured on the industrial scale in the fermenter. So these fermenter are large bags. They can be kept at the optimum pH and temperature for the Fusarium to grow. Okay, Fusarium is another kind of fungus, just like penicillium. Penicillium also is kind of fungus. So that's why Fusarium will be another family of the fungus. So this fungus is grown in the aerobic conditions and it is provided with the oxygens and provided with the glucose syrups. These are syrups as a food source to allow the fungus to respire. Same to the bacteria just now, okay? You need to provide the fungus with the aerobic conditions. And then you need to provide it with glucose, okay? So glucose become their food source. So that's why the fungus, they can do what? They can do the aerobic respirations. So the fungus just grow and multiply within the fermenter. Yeah, so that's why the fungus biomass, they will harvest it and then they purify to produce mycoproteins. Because this fungus, which is this Fusarium fungus, they can produce the mycoprotein. And then you want to get these mycoproteins. So that's why you need to grow the fungus. So the fungus, they will reproduce in a large number. So when they reproduce, right, they can produce a lot of products, which is the mycoproteins. Okay, mycoproteins is a protein-rich foods suitable for the vegetarians, like used in corn products. So this is the veggie, okay, vegetarians, they will like this kind of mycoproteins because vegetarian, right, they cannot eat vegetarian, they cannot eat the meat. Meat is a source of proteins, right? So that's why they cannot eat the meat. So that's why they need to find an alternate source of proteins. For example, they need to find the proteins from the plant. You have a plant-based protein. So that's why mycoproteins is a proteins produced from the fungus, right? The fungus. It's not for the animal. So that's why it can be one of the alternative source of proteins. So the productions, the products which is they use this mycoprotein, right, is a corn, okay? Corn. And then now we talk about the insulin, yeah, which is just I talked to you for the insulin. So insulin, they using what? Okay, the genetic modifications. Genetic modification known as what? 基因改造. You are actually modify the genome material. So genetic modification is they are referring to the changing, okay, changing in the genetic material of the organism by removing or altering the genes with that organism. So they actually do this by inserting the genes from another organism. So the organism receiving the genetic material are said to be genetically modified. We call it what? GMO. GMO. So that's why whenever you see the product label GMO, you need to be very aware. So because this GMO means what? Genetic modified products. GMO or genetic modified organism. So they are described as a transgenic organism. Transgenic refer to because inside that organism they contain the foreign genes isolated from other living organisms. So the DNA of organisms, they now contain the DNA from other organisms. They are known as recombinant DNA. Okay, so the genes for the human insulin have been inserted into bacteria. They will produce a large amount of human insulin. So later they can be purified to treat diabetes, 糖尿病. Okay, the end. Okay, few more example. What's meant by genetic modifications? You're actually changing the genetic material of the organism by removing, changing, or inserting the individual genes from another organism. And then example, okay, which is the products produced from the genetic modifications? It will be the human insulin, okay, human insulin proteins, and also some of the crop plant, such as wheat and maize. So they can, the scientists, right, they can do the genetic modification on that plant species of the wheat and maize. So that's why they will modify them, okay, to contain the genes from the bacteria. So which is they refer to, they will isolate the genes from the bacteria. So the genes, they able to have the resistance against the insecticide or any kind of pesticide, something like that. So they just extract out the genes, which is they carry the pesticide or insecticide resistant characteristic. So they just insert the genes back into the plant cell, and then they'll just put back into plant. So that's why the plant now they become transgenic plant, because they consist of foreign genes from the bacteria. Okay, so this is how to do the genetic modification on the wheat and maize. Okay, so they just contain the genes from the bacteria. They produce the poisons. They kill the insect, making them resistant to the insect pests such as the caterpillar. And then crops have been genetically modified to make them resistant to certain herbicides. Okay, herbicide refer to any kind of drugs. They just spray on the crop, right? It will kill the weeds. And then crop has been also produced genetically modified to produce additional vitamins. We call it golden rice. Actually, this one you can refer to here. Okay, you can refer to this one. Oh, let me show the example. Yeah. So usually the exam they will ask in this way. Okay, they just something like ask you to state. Okay, state few example of the genetic modified. Ask you to state few example of genetic modifications products. So you can say insect resistance in the crop plant, or the herbicide resistance in the crop plants. You can say, so what's the advantage? So insect resistance is they can produce a poison, okay? They can kill the insect pest. And then herbicide resistance is they can be resistant to any kind of herbicides. So that's why you spray the you can spray the crops, okay, the transgenic crops with the weed side. The weeds, you call it 杂草. So that's why only the herbicide will kill the weeds. It will not kill the crops. Because usually the herbicide is very, very strong. The effect is very harmful. So there's a lot of time they not only kill the weeds, but they also kill the crop as well. So we don't want to it happen. Yeah, we want to save the crops, but you want to eliminate the weeds. So that's why you will the scientists, right, they will come up with this herbicide resistant plant, which carry the gene resistant against the herbicide. And then for this one, they can just improve the nutritional quality or in the rice. So yeah, so what's meant golden rice? Golden rice, when the rice from the appearance, it will appear golden yellow instead of white color, okay, which is the normal white rice. Why? It's because the golden rice are being enriched with the vitamin A. See, this is a vitamin A. Vitamin A is a kind of lipid soluble vitamins. 油溶性的这个 vitamin lipid soluble, so that's why it will make the rice appear become yellow. So that's why you can prevent any kind of nutrition deficiency disease caused by the vitamin A. So this is a benefit for the genetic modification and their example. So this process just showing about how you're able to do the genetic modification by using the human proteins. So just remember about a few important point, okay? Registration enzyme to cut that particular of the gene. So after you cut them, you can produce a sticky end. Now, so what makes a sticky end? Sticky end, they refer to a short sections of the unpaired base. So that's why we call it sticky end. So that's why a bacterial plasmid is cut by the same registration enzyme, leaving it with the corresponding sticky ends. So use the registration enzyme to cut on the bacterial plasmid to get the sticky end. And also you use the registration enzyme to cut on the gene. Okay, the human insulin gene also on the chromosome. Okay, on the chromosome. So that's why here is a registration gene. So they just cut part of genes. So that's why they can form a sticky end. So this sticky end they refer to the unpaired base. Okay, unpaired base. So the plasmid and the isolated genes are being joined together by the DNA ligase. So that's why you need to remember these two enzyme names. And then they will ask it, registration enzyme, which is just like scissor, because they just cut the portion of the gene and then to produce a sticky end. Then how about DNA? DNA ligase just like glue, because they can join the sticky ends together, become one DNA. Okay, they just joined by the DNA ligase. So if there's two pieces of DNA have matching sticky ends, one from the bacterial plasmid, one from the human insulin genes. Okay, because they have been cut by the same registration enzyme. So that's why DNA ligase, they will link them to form the single unbroken molecule of DNA. Okay, so that's why they can transform them, become recombinant plasmid. Because they already joined the human insulin genes, the sticky end for the human insulin genes into the plasmid vectors. So by the DNA ligase, you can fuse them together, become recombinant plasmid. So later you can insert this recombinant plasmid into the bacterial cell. Okay, so that's why genetic engineered plasmid is inserted into bacterial cell. So when the bacteria they do the asexual productions, they can copy the plasmid very well. So that's why they can allow the recombinant plasmid to quickly be spread. As the bacteria multiply, they will express all the genes and then they will make the human proteins. So you also can place this genetically engineered bacteria in the fermenter, which is a biofermenter. So that's why you can produce a lot of the products, okay, which is the human proteins. Okay, for this one, you take a note. You take a note here. Okay, why bacteria is very useful? Because there's no ethical issue if you want to make their growth. Okay, because bacteria, right, not like in the animals, they will feel pain and distress. But bacteria just bacteria, right, and then very, very small. Then bacteria, they have a plasmid, which is only found in bacteria. Yeah, so it will make them become very easy to move and manipulate by inserting the genes into the bacteria. By inserting the gene into the plasmid DNA, and then they put that into the bacterial cell. So bacterial cell, they can read all the genes, and then they can produce the proteins. So it's just very, 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 very convenient for them. So bacteria is just like the medium, okay? A medium help to produce the proteins from the foreign gene insert to them. And then another thing is bacteria itself, they contain the same genetic code as the organism we are taking the genes from. So they can easily read and they can produce the same proteins. Okay, so this is by what? And also, let me ask you, our DNA have 4 types of base, which is the A, T, C, G. And then do you think this A, T, C, G also found in the bacteria DNA? Or the bacteria DNA, there will be another form of gene, another form of nucleotide base sequence. What do you think? In the bacteria, they will have a same nucleotide base sequence, which is the ATCG like the humans, or it will not have the same genetic sequence.
Not the same.
Same. Okay. Yeah. The answer is same. Yeah. So that's why the ATCG, right, which is adenine thymine guanine cytosines, we call as what? Universal genetic code. It's universal. It's same for all kind of living organisms. In the humans, in the animals, right, they also being formed by this 4 type of base, become their DNA. Just the difference in the sequence, okay, the sequence, but the base is the same, just same 4 type of base. So that's why even though we insert the human insulin genes into the bacterial plasmid, right, and then you insert back to the bacteria they can still read the code. They're able to read the code because they are derived from the same nucleotide base sequence. Same nucleotide base, which is a 4 type base. Okay, so here's a thing that you need to write in the answer if the question will come out this. Okay, the genetic modified crops, right? You need to know what the advantage and disadvantage are. So this one you just have a read. Okay, so usually the advantage is because you will produce the GMO, okay, the genetic modified crops, and then this crop, right, is usually resistant towards the insecticide or herbicide. They can have a higher yield of productions. So this is the advantage. Okay, increase yield from the crops. Okay, because they are not competing with the weeds for the resource. And then they can reduce use of chemical such as herbicide and pesticide because it's now already being resistant to the herbicide and pesticide. So that's why it will produce a GM crop which is much more stronger compared to the normal crop. And then what's the disadvantage? The disadvantage is by this way, right, you can actually increase the cost of the seeds. Okay, so some company they just make the GM seed charge more than the normal seed because they need some time and a lot of resources to develop them. So that's why they will cost more expensive. So that's why this means a smaller, poorer farmer, they cannot compete with the larger farmer because they don't have the budget to buy that seed, the GMO seed. And then you are increased dependency on the certain chemical such as a herbicide. So they are crop resistant too. Okay, so this is mean by what? This is a disadvantage. Okay, you are increasing the requires a dependency on the chemical such as a herbicide. So the crop are resistant to. So often made by the same company, they produce seeds and more expensive to buy. So this is referred to because if you produce a GMO crop, and then these GMO crops, right, they will have we call it more resistance to the herbicide or insecticide or any kind of chemical products. Okay, so that's why you will pass these resistant genes to the next generations. If you're able to make these GMO crops with another plant, you pass this resistant gene, okay, to the next generations. So this resistant gene, right, is not really good for them, okay, because it will develop resistance, okay, it will develop resistance, just like the antibiotic resistance. And then your risk of inserted genes being transferred to the wild plant by pollinations, which could reduce the usefulness of the GM crop. Because the weeds, they can also gain the genes, okay, gains the foreign genes, and then they make them resistant to herbicide. Okay, so this is not good for them because you just want to target on that specific range of the GM crop only, but you want to avoid the resistant genes being transferred to the wild plants. Because if the resistant genes, okay, resistant gene is referred to the genes resistant against the herbicide, insecticides, any kind of chemical products. So if these genes, right, being passed to the wild plants, so now the wild plant, they carry the resistant genes. So now the herbicide, insecticide, unable to kill the wild plant because they already have the resistant genes. So this will be a disaster, okay? Because you cannot kill the wild plant. Yeah, even though even if the weeds cause what 杂草, they will gain the genes, right? So that's why it's very hard to kill the weeds. And then you're actually reducing the biodiversity as there's fewer plant species when the herbicide has been used. So this will cause an impact to the insect and the insect eating the birds. So by this way, right, you are playing gods, you know, because this GMO, okay, just something like natural, something like the artificial selections, okay. But in the artificial selections, you didn't modify their genes. You just select, okay, with the trait that you want and make with another organism, and then you produce a desired product that you want. But in this GMO, this genetic modification, right, you are actually modify the genes. So that's why you modify the genes and then you produce that organism. So the organism will carry the genes, the modified genes. So that's why you are actually reducing the biodiversity. Okay, because you are actually producing the fewer plant species. You just only focus on that plant species. Okay, so some research also show that the plant they have had the genes inserted to them do not show as well as a non-GM plant. Okay, this is not too important. Okay, show that even though the plant they carry the GMO gene, okay, they carry the foreign genes. So, but their performance just like in the normal plant. So this will lose their value because we expect that the GMO plant they will perform better than the normal plant. Okay, so in the exam, right, you just need to know about this. Okay, increase yield for the advantage. You need to remember about 2 points. Okay, at least 2 or 3 points. Increase yield, reduce use of chemical, and then the disadvantage is they become more expensive. Okay, more expensive, and then you are reducing the biodiversity, and then you increase dependency on the chemical such as herbicides. And then you cause a wild plant also level resistant, which is not good for them. Yeah, we finished. Okay, so at this point, we already finished the total 21 chapter for the IGCSE. And then let me show some of the questions, the example of question, okay? So for this one, right, this one, the important keywords, okay, I already upload in the Google Classroom, and then remember to refer it out when you do the revisions. Yeah, because this is actually based on the syllabus, okay, syllabus in the IGCSE. You need to ensure that you are know about the syllabus. 你要了解那个课程是包括什么内容. So that's why you just read on the syllabus. And then you ensure that you already know how to answer the questions on the syllabus. So this is you can read by yourself. And then I will show you about what? I want to show you about the so you can see that here's the fruit productions. I mean the fruit juice productions. See, the apple juice, right, initially is very cloudy. So after you apply it with the enzyme pectinase, you produce a clarified apple juice. Okay, let me show this one. Yeah. So this example of the question asking in the biotechnology and the genetic engineering. So is see, you are putting the flour, salt, and water into the bowl, and then you mix them. And then organism A, you produce the organism A, and the energy source are being added. Yeah, so step by step, right? So this is actually an example of the yeast fermentations in the bread making process. Okay, so see, name of the source of energy used by the A. Okay, so what is the source of energy supplied to the organism A? Which is the glucose. So just like glucose is a common source of energy. Okay, so the function of glucose is they're just acting like the source of energy because they can do the cell aerobic respirations by using the glucose. To break down the glucose, release the chemical energy. Okay, then state the name of process in the step 3 that cause the air bubble to form in the dough. So yeah, I see air bubble inside the dough, right? Refer to they already do the respirations because they produce the ethanol and the carbon dioxide. So carbon dioxide is a gas. Okay, explain for different temperature used in step 3 and 5. 3 and 5 refer to what? Here. Why in the step 3, right, they use 35 degrees Celsius, but why in the step 5, they use about 200 degrees Celsius? Why? So this is their different purpose. Okay, but in the step 3, you lowering the temperature down to about 35. It's because you want to provide the optimum temperature for the growing of the enzyme, because enzyme prefer optimum temperature to grow. And then step 5, okay, the high temperature will kill the yeast to evaporate the ethanol. Because later, after you mix the reagents, right, including the flour, salt, water, and the yeast, and then you produce a lot of ethanol and the carbon dioxide. So later you need to isolate your product out, okay? So before you isolate your products out, right, you need to bake them, okay, for about 200 degrees Celsius. Okay, why? It's because this 200 degrees Celsius is a high temperature, okay? This high temperature, they can kill the yeast and evaporate the ethanol. Because you want to ensure the fermentation process, right, being stopped. You want to ensure them being stopped. So that's why you bake them with the high temperature, so you kill all the yeast. Okay. And then explain why yeast is used in the bread making process is because the yeast, they can do the anaerobic respiration, so they can produce the carbon dioxide. Yeah, they will cause a bread to rise. And then yeast, they will produce an enzyme. Okay, yeast will produce an enzyme. You no need to know about the name of enzyme, just need to know it's an enzyme catalyzed reaction. So that's why you need optimal pH and optimal temperature supply to them. And then for this one, okay, describe how penicillin, okay, the fungus is being used to make the antibiotic penicillin. So the fungus penicillin grow in the fermenters. So they will do the aerobic respiration. So when they do aerobic respiration, they produce a large amount of the carbon dioxide, water, and the energy. So the carbon dioxide is bubbles, okay? See, air is bubble through. So you can see, right, there's a bubbles inside the fermenter because due to the production of the carbon dioxide by performing the aerobic respirations. So they'll provide the sugar and amacid also. Okay, so the sugar and amacid, which is a nutrient medium supply to the penicillin. So that's why the penicillin, they can use those nutrients for growth and the reproductions. Okay. Yeah, for the growth reproductions, because tissue growth require amino acids. Okay, and then when the tissue, they require the energy source they need from the glucose. And this one, water jacket. Okay, this water jacket, right, they are referred to just now. Okay, in the picture, they're showing the water enter inside and then the water flow away. So this causes water jacket. So water jackets, okay, ensure the cool water pass through the medium. So they want to ensure the temperature are being maintained optimum. Because they want to just remove heat away from the fermenter. Because during this fermentation process inside the fermenter, the biofermenter, they will produce a lot of heat. And then it is enclosed, so that's why they will cause a lot of heat being trapped inside. So once the heat being trapped inside the fermenter, it will cause rising the temperature. So it will affect the enzyme, the activity of enzyme. So you want to prevent this happen. So that's why you need to develop the water jackets to ensure the cool water flow through the beaker, flow through the medium, right? And then you can at least reduce the temperature. Why antibiotic can be killed to treat the bacterial infections? Because virus itself are not alive, and then virus do not have cell wall. Okay, so at this remember, virus is not being killed by antibiotic because due to a different component. Okay, in the virus, okay, I hope you still remember. In the virus, we have what kind of structures? And those in the virus, right? Virus have nucleus or not? Do you think virus have nucleus?
No.
No, yeah. Virus, they don't have nucleus. Inside, they only have DNA. They all have DNA and the capsids, okay? So they'll be in this kind of structures. Yeah. So this is the capsid, okay? Capsids are protein coats. And then they will have a DNA inside. Yeah, DNA inside. And then yeah, so just in these two components. And then they will be surrounded by the envelope. So this will become the virus structures. Envelope, which is a protein envelope. Okay. Treat by the insulin, it will be diabetes. Okay, so this one also to describe the process of using the bacteria to produce the human proteins. 5 marks. So that's why you need to know about the process. You need to know about the two enzymes, okay? The restriction enzyme. So you are actually using the restriction enzyme to cut the human gene and also the also the plasmid DNA from the bacteria. You form a sticky end. Okay, so later you use a DNA ligase to join the plasmid and the gene together, becoming a recombinant DNA. So later the plasmid will insert back to the bacteria. Bacteria will reproduce and they will produce an insulin. Okay, so the steam kill the fermenter. So that's why you are actually use a steam to kill the pathogens and prevent contaminations. Yeah. So what kind of three conditions? Okay, the three conditions inside the fermenter have been measured is pH, temperature, oxygen, and the nutrients. Okay, so those requirements need to be fulfilled so you can able to grow the large amount of the products that you want. And then for this one, okay, why is it important to stir the contents continuously? Because you want to allow the even distribution of content inside. You want to allow the content inside being mixed uniformly. Okay, so and also you can prevent clumping of the content. So that's why the bacteria, they can absorb the substance more easily. So that's why they can grow better. And then the advantage, okay, this will be another questions, okay, come out in the exam before. The advantage of the GMO plant, okay, GMO crops is, see, you have the disease resistant, drought resistant, insect resistant, herbicide resistant. Yeah, produce faster yield, higher yield. Yeah, you can increase the income. So this is just an advantage, yeah. Okay, advantage. So that's why this kind of resistant genes being passed through the wild plants, right? It will be disaster for them because you cannot kill the wild plant. Okay, explain the evidence suggests that bacteria in the dish F are resistant to the penicillin. So how you able to recognize this one? Okay, how you able to look this one is, this is the bacteria plate. So this is a petri dish, right? Petri dish is referred to the plate. So let me show you. Maybe you first time see this. We call it a petri dish with the bacteria. Okay. They will use a dish, right? This is we call it what? Medium. Okay, the neutron media. This one is neutron media. So this neutron media, they'll be supplemented with the nutrients. Okay, so whenever you just grow the bacteria on it, so you can see that the bacteria, they can grow well on the media. Okay, they can just grow well on the media. So in future, if you study microbiology, you can have a chance to grow your own bacteria in this petri dish. It will be interesting. Yeah, so that's why what happened is, okay, suggests that the bacteria in the dish F are resistant to penicillin. So you can see you have about 2 plate. Okay, 2 plate. And then the middle part will be the penicillin. See here, okay, here will be penicillin. And then at those one, you can have a guess. You think which one already developed to the penicillin? Is E or F? Which one, which penicillin in the E or F already developed the resistance to the bacteria? So this is the question. E or F?
E.
E? You think it's E, okay? Penicillin, you can just imagine that penicillin is just grown in this plate, right? So if they already develop resistance to the, okay, maybe I can just rephrase in this way, okay? Better for you to understand. So you can see the small clear area, okay? So the middle part, okay, the middle part is a paper disc soaked in the penicillin, which is the antibiotic, okay? It's antibiotic on the paper disc, on this central part of the paper disc. And then this is the antibody, right? So that's why they can kill the surrounding bacteria. So whenever they kill the surrounding bacteria, they will produce a clear zone. So this is a clear zone, okay? You can see this black color, right? This is a clear zone. Clear zone without any bacteria growing. So as you can compare this E and F, okay, you can see the bacteria, right? This penicillin, okay, they can kill the bacteria in the plate E. But this penicillin in the plate F, right, they look like they're unable to kill more bacteria. So because they can produce a small clear zone only. So they refer to in the F, right? In the F, okay, in the F, they have a small clear area. So the F, which is a penicillin in the plate F, in the dish F, right? They unable to kill the bacteria. Okay, penicillin in the plate dish F, unable to kill the bacteria. So this means what? The antibody not able to kill the bacteria means that the antibody already developed the resistance. Means which refer to the bacteria. Okay, this bacteria here. The bacteria, they already to develop resistance. Develop resistance towards the antibiotic. Okay, the penicillin. Okay, in the plate F. So in the plate E, right, the bacteria still susceptible. Okay, susceptible refer to they can still being killed by the antibiotic in the plate E. Yeah, still susceptible. Okay, and being killed by the antibiotic penicillin in the plate E. So the bacteria which develop resistance is in the plate F, because bacteria they can just grow, they can still grow around the penicillin because penicillin cannot kill them. So that's why bacteria they already develop resistance. Okay. I think I need to show you what's meant by antibiotic resistant bacteria. Yeah, you know, antibiotic resistant bacteria, this one, antibiotic resistant, which is referred to 抗药性, you know, 抗药性. Yeah, and then this is a common issue in the medical field or in the healthcare field. So explain how the bacteria become resistant to the antibiotic. How the bacteria develop a resistance to the antibiotic, and then how the human they can reduce the issue of the antibiotic resistance. So how it become resistant is because bacteria, right, they grow in the large populations. Okay, and then this large population of bacteria, they will have difference in their genetic compositions. So they will have the mutations. Okay, they will develop mutations. What's my mutations? Mutation is they will produce a new alleles. Okay, new alleles. Yeah. So it's like mutations in the bacteria, they will have the resistant genes. So it means that only the bacteria are being mutated. 突变了, 基因突变了, they will have this kind of resistant genes. Other kind of normal bacteria, which is not undergo mutations, right? They will not carry this resistant gene. Okay, so mutation refer to the change in the DNA-based sequence. And then at those, what's the source of mutations other than radiations? Okay, if the question asks you state about 2 source of mutation, radiation also, and then you can use what kind of the source of the mutations. You can talk about the chemical, okay? The chemical products are, and then you can talk about the UV rays, okay, the ultraviolet rays. So they can contribute to the mutation happen. So bacteria with the resistant genes, they can survive and reproduce. And then they can pass on the resistant gene to the offspring. So this is very important keyword. See, survive and reproduce. Pass the gene, the resistant genes to the next generations. So it's like bacteria with not resistant gene, they would die. Okay, so that's why it will cause the progeny of the bacteria. They will carry more and more resistant gene. Because only the one who carry the resistant gene, they can survive and they can reproduce. So this is the conditions, okay, and this is of course phenomenon for the natural selections. So it means that the bacteria which able to develop the resistant gene against the antibody, right? It is a process belong to the natural selections. Yeah, so how you're able to reduce the problem is the doctor, okay, the doctors or any kind of healthcare professional, they only can use the antibiotic when it's essential. Means that antibiotic can only be used for treatment purpose, not for the preventions. Okay, because in future you learn about the treatment of disease and prevention of disease is different thing. Okay, and then why the antibiotic is overused? Because over abuse of the antibiotic. Okay, because whenever you visit a doctor, right, even though you don't have that bacterial infections, but the doctor will still prescribe you with the antibiotic. So I think at this point you eat a lot of antibiotic before, right? Whenever you see doctor will always prescribe you with antibiotic. Because antibiotic just only treat for the bacterial infections. Especially for the secondary bacterial infections. So that's why you cause an over abuse, okay, which is overuse of antibiotic. So it means that in your conditions, actually you don't need to use antibiotic, but the doctor still prescribe you antibiotic. So that's why it cause an over abuse of this drug. So this will encourage the development of the antibiotic resistant bacteria in your body. So you can reduce the problem is you only use the antibiotic when it's needed, when it's essential. Okay. And then whenever you eat the antibiotic, right, you need to finish the whole antibiotic treatments. You need to complete the full course of the prescribed antibiotic. Okay. Which is you need to finish all the antibiotic, okay, not eating half and then still half of the antibiotic not being finished. So it will be not good because this will encourage the development of the antibiotic resistance in your body. And then you need to isolate the patients with the infections, improve healthcare and hygiene and cleanliness. Okay, because when you improve the healthcare or hygiene, right, you can reduce the infection, especially bacterial infection, effectively, seriously. So whenever you can control the bacterial infection by improving the hygiene, so why is there need to use antibiotic to control it? Okay, and then why bacteria are useful in the antibiotic? They can reproduce rapidly. They have the plasmid. They can do the genetic modifications. There will be no ethical issue used to make the complex molecule. Okay, so this is about it. So in the exam question, right, they will ask you to recognize, okay, which one is a DNA ligase, which one is a registration enzyme, and then they will produce what? Recombinant plasmid or sticky end. So those are keywords you need to know. Okay, so they just saying, okay. Maybe you are wondering, okay, why the sticky end from the bacterial plasmid they can combine or they can join with the sticky end from the human gene? It's because the sticky end of the human gene is complementary to the plasmid. So later the DNA ligase, they can join them together, become the recombinant plasmid. And then explain the role of mRNA in the bacteria. mRNA, they are being used in what? Transcription process, okay? mRNA is being involved in the transcription process, which is whenever the DNA, they need to, they will unwind, okay, expose their base sequence to the mRNA. So later the mRNA, they will carry the genetic information from DNA and then they will go out from the nucleus, enter to cytoplasm. So see, mRNA, they can move through the cytoplasm from the nucleus and then they will move to the ribosome. So later, ribosome, they will just assemble. Assemble means that they will join the amino acid together, become the proteins, okay, become protein. So that's why the sequence of base in the mRNA, they can determine the orders of the amino acids. Okay. So see, the question is actually repeating. Okay, it's actually repeating. Okay, that's all. Yeah. So for the homework today, okay, that's well. I will start to give you do the actual, the actual exam questions from 2024 to 2025. Okay, especially these kind of questions. Okay, you need to complete it. If you don't have the time, okay, I understand you still have other subject to study, right? If you don't have time, but you still need to try at least 2 or 3 questions, please. Okay. I think you can at least try 2 or 3 questions, and then this is some of the example of question, okay, which is being come out in the exam before. For example, right, so this is how the actual exam will ask you. Let me show you one of the question. See, at least you need to read the questions. But after you read the question, right, you need to question and answer like this, okay? Read the answer and questions, and then you need to try to do it, okay? At least try 2 or 3 questions, okay? Because I just worry that you read the question, you didn't try by yourself, you didn't remember it, and then you cannot answer the question in the exam. Because IGCSE is not as hard as you think. Some of the questions will be repeating again. So it's like it's very important you need to study the past year questions. Yeah. Okay, so I think I will do the revision for you for the paper 4, especially for paper 4, okay? And also the paper 6, okay? And also paper 6, I will find the time to discuss with you. Yeah. So later I will send you some of the MCQ, okay? Multiple choice question, which is paper 2 for the 2024, 2025, and then you can have a try. And then you send back to me, I will mark for it, okay? Yeah, so this is example of the question. Yeah. See, they come out this kind of question, okay? Fermenter. So what kind of organism they can produce a penicillin? So it's a fungus. Fungus is a kind of penicillin, okay? So describe how fermenter can be sterilized. So that's why you use a steam, okay? Steam under a high temperature to sterilize. Or can use a UV radiation or gamma rays. Yeah. And then you need to label it. See, just need to label it. So the paddles is to just keep them in uniform. Nutrient you need to supply the glucose. Probe to like monitor, okay, how to monitor any change in the temperature and the pH. So after, what happens if the liquid containing the penicillins, after it's been collected from the fermenter, so later they will undergo the purifications. So purification is a process which is, they will just remove the impurity in the penicillin. Impurity refers to the dirty substance in the penicillin. So that's why they just want that penicillin product. So they need to undergo purifications. 净化, okay, refer to the 净化. And then for this one, so you can see, right, the 3 nutrients are inside the milk, the proteins, lactose, and calcium. So they ask about what role in the body. Yeah, so the function of protein is they help in the tissue grow or make new cell. And then the sugar, okay, sugar, no matter it's lactose or glucose, right, the function is they become the source, okay, source of energy. So they can provide them, I mean, they can like use the lactose or the glucose to do the cellular aerobic respiration, so that's why they can release the energy supply to the body cell. And also the question, right? Usually in the IGCSE question, they will combine about few topics together, become one question. So in this question, right, you can see some of the question is come from the chapter 7. Okay, some is come from chapter 21. So that's why you cannot skip one of the chapter. You need to study all, okay? No choice, no choice. But you can focus on some important topics, okay? So later I will send you the important topics. So any kind of questions, which is very interesting. Yeah. So see, explain why lactose is being not absorbed by the small intestines. Because there's no enzyme to digest the lactose, no lactase. So lactase is a disaccharide, okay? They cannot directly absorb into the intestine villi. So just remember, your intestine villi only able to absorb the monomer, which is the glucose, the amino acids, or the fatty acids. Sometimes they'll ask about diarrhea. Okay, so I think this one you can read by yourself. Okay, you can read by yourself. Just need to know, okay, what kind of keyword that you should write. So those are the keywords. I see. So here is a question just now, okay, 2018. Yeah, so that's why the bacteria in the dish F is resistant, because there's no clear area. No clear area refer to the bacteria already develop resistance to the antibiotic. 细菌已经对这个抗素有那个抗药性。So antibiotic cannot kill the bacteria. Yeah. Okay. So almost all the questions are already repeated. Okay, repeated. So that's why it's very important. You need to study the past year. So you need to know how to answer. Okay. Yeah. So I think that's all for today. And then I send some of the homeworks in the groups, and then you need to do it. Okay. In the next class, right? And then you need to submit to me before the next class. Yeah. Okay. Happy study. Remember to read the this one. Okay. I will upload this in the Google Classroom. This one, the quiz card for each chapter. And then when you read, right, you need to be aware for this one. Okay. The question I put a star. Okay, because this one means that it come out in the past year paper before. Okay, you need to ensure that you need to know how to answer the question, especially for the star question. But other kind of question you can read also. Okay, you can read also. Okay, that's all for today. And then do you have any question to ask me now? Okay, yeah. Okay, happy study. Bye.
Thank you.
Bye.